Welcome to this explainer, everybody. I'm your host, Dr. Chem, and today we're jumping right into what I think is a truly epic biological investigation. Just picture this, a pristine laboratory bench, bathed in soft, dramatic, cinematic lighting, and right in the center, an ordinary, fresh potato. Honestly, it looks completely unassuming, right? But beneath that skin is a bustling, microscopic world governed by completely invisible forces. And these are the very forces that literally dictate life and death for cells. So let's unpack exactly how this all works. Now, you might have pondered this exact question in your own kitchen. Why is it that fresh vegetables, like cucumbers or potatoes, suddenly get all limp and shrink when you sprinkle them with salt? Or, think about this, why do you get so incredibly thirsty after eating a super heavy, salty meal? It kind of seems like just a simple culinary reaction, right? But it's actually our very first clue in solving a massive biological mystery. It all comes down to the invisible movement of water. And this phenomenon, it's happening inside you and inside our potato right this very second. So, that invisible movement actually has a name. It's called osmosis. The formal scientific definition is the net movement of water molecules from a region of higher water potential to a region of lower water potential, right through a partially permeable membrane. To really wrap our heads around this, imagine zooming way in on this cellular gate in stunning 3D. It's incredibly picky. It's not going to let large sugar or salt molecules pass, but it does allow these glowing, bright blue water molecules to just float right through. Basically, the water naturally wants to balance things out, moving from where it's abundant and pure to where it's crowded with other dissolved particles. And looking at a side-by-side comparison really brilliantly illustrates what happens when we manipulate that flow. On one side, you've got a plump, perfectly healthy plant cell. It is just packed with water, swelling up like a water balloon, and pushing firmly against its rigid cell wall. We call this a turgid cell. But on the other side, well, it's a completely different story. This cell has basically been robbed of its water. It's totally crumpled, and the delicate inner membrane is literally peeling away from the cell wall. This process is called plasmolysis, and honestly, the cell looks completely collapsed. So these observations build into the fundamental rules of cellular environments, which we call the 3 states of tonicity. Think of it kind of like the biological version of the Goldilocks rule. In a hypotonic environment, there's way too much pure water outside the cell, so water just rushes in. Now, in a hypertonic environment, like our salted vegetables, there's too little water outside, so the cell's water rushes out to dilute the salt. But what every living cell constantly seeks is that 3rd state, isotonic. This is the just right environment, where the water potential inside and outside the cell is perfectly balanced and water just moves back and forth at an equal, harmonious rate. All right, talking about invisible glowing molecules is great and all, but here in the lab, we demand evidence. So let's step back to the bench to actually prove this invisible theory. We're going to catch osmosis in the act, and we're going to do it using nothing more than carefully prepared strips of our ordinary potato and a few neatly arranged beakers of liquid. Now, the absolutely crucial point here is that to truly observe this phenomenon, we have to be absolutely ruthless with our scientific method. Notice how we can't just casually hack up a potato. No way. We use a specialized tool called a cork borer, so every single potato cylinder has the exact same diameter. Then we use a scalpel to trim the ends completely flat to ensure equal surface area. They're all going to stay submerged for exactly 30 minutes. And, critically, we'll use the exact same gentle blotting technique before weighing them. We are literally eliminating every single variable except one. So here's the game plan for our experiment. Step 1, cut those highly uniform potato cylinders. Step 2, we place them on a highly sensitive digital balance to record their initial mass, right down to the decimal. Step 3, we submerge them into our labeled beakers. And here's where the suspense really builds. We have a range of sucrose, or sugar, solutions, starting from 0.0, which is pure distilled water, all the way up to a highly concentrated 1.0 moles per cubic decimeter. Finally, step 4. After 30 minutes, we gently blot them dry and weigh their final mass. So what do you think is going to happen to the potato in the pure water versus the one in the heavy sugar? Okay, the results are in. Look closely at the raw laboratory data we just captured. These numbers are wild. The potato cylinder submerged in pure water started at 3.12 grams, and it actually gained mass, ballooning up to 3.40 grams. Water literally forced its way into the potato's cells. But jump down to that 1.0 strong sugar solution. It started at 3.11 grams and dropped all the way down to 2.76 grams. That hypertonic sugar solution violently sucked the water right out of the potato. And then, right there in the middle, at point 4, the mass started at 3.15 and ended exactly at 3.15. But hold on, before we go declaring victory, we need to ask a highly critical, analytical question. We've got our raw mass changes, sure, but why in the world of biology must we calculate the percentage change in mass rather than just using the absolute mass change? Like, why not just say, hey, it gained 0.28 grams and call it a day? Why is that raw number just not good enough for our investigation? Well, it's because mathematics is honestly our greatest narrative tool here. You see, even with our precision cork borer, it is physically impossible to get every single potato cylinder to the exact same starting milligram. A cylinder that starts at 3.50 grams and gains 0.28 grams is undergoing a very different biological stress than a tiny cylinder that starts at 2.80 grams and gains that exact same 0.28 grams. So by dividing the change in mass by the initial mass and then multiplying by 100, we calculate the percentage change. This beautifully levels the playing field, completely normalizing the data so we can fairly compare every single potato cell's journey. And when we plot that percentage change on a chart, we truly visualize the invisible. Just look at the dramatic downward trend here. On the far left, in pure water, we see a massive positive spike of plus 9.0%. The potato cells are completely turgid and swollen. But as the concentration of the surrounding sugar solution increases, the data drops lower and lower, plunging deep into negative territory. Down at that 1.0 concentration, we're looking at a negative 11.3% mass loss. Those cells are flaccid, they're crumbling, and they are fully plasmolyzed. But the absolute star of the show, the grand reveal of our entire experiment, is this exact number right here, 0.4 moles per cubic decimeter. This is the isotonic point. If you trace the line tracking our potato's mass change, 0.4 is the exact moment the graph crosses the 0% line. At this specific concentration, there is literally zero net movement of water. Those glowing blue molecules are entering and exiting the potato cells at the exact same rate. We have basically just used simple math to successfully estimate the exact internal solute concentration of a living organism. How cool is that? All right, let's zoom out from the lab bench for a moment. Because understanding osmosis isn't just about passing some biology exam, it actually dictates the entire world around us. So why does this matter? Well, first off, plant posture. Without bones, plants rely entirely on the turgor pressure of water inflating their cells to reach for the sun. Second, food preservation. For centuries, humans have packed meats and vegetables in heavy salt. Why? Because that hypertonic environment draws all the water out, making it impossible for decay-causing bacteria to survive. And third, human hydration. When a person is dangerously dehydrated, doctors don't just give them pure water. They administer an oral rehydration solution, a precise isotonic balance of salt and sugar to safely coax water back into human cells without bursting them. I really want to hammer this point home with a profound thought from our source material. Without the stiffness of the plant cells, the plant would fall under its own weight. Just picture a towering redwood tree or a massive, vibrant sunflower. They're quite literally architectural miracles, standing on microscopic pillars of water. The sheer hydraulic pressure generated by osmosis inside those tiny, selectively permeable membranes is actually strong enough to defy gravity itself. Which leaves us with one final, deeply provocative thought to wrap up our explainer today. We've just seen a potato crumble and lose over 11% of its mass when exposed to a hypertonic, salty environment. But remember, your body is made of cells too. And yours don't even have rigid cell walls. They are fragile, and they desperately rely on that perfect isotonic balance. So knowing what you now know about the ruthless, completely invisible laws of osmosis, what exactly do you think happens to the cells in your own throat and blood when you're stranded in the ocean and you take a huge gulp of hypertonic seawater? I'll let you ponder that fascinating, slightly terrifying biological reality. Keep exploring, keep questioning, and I'll see you next time.